Hello, everyone, and welcome to the 13th Field Notes live event. My name is Casey Latta, and I am the MLRA Soil Survey Office Leader in Lincoln, Nebraska, and the Field Notes Regional Representative for the North Central Soil Survey region. As a regional representative, I serve on the Field Notes Review Committee. The Review Committee solicits and selects topics for each webinar. We have selected two exciting topics for you today, and we encourage you to ask questions at any time using the Q&A panel. The Q&A panel should open by default. However, if for some reason your Q&A panel is not open, simply click on the question mark icon located on the right side of your screen. For closed captions, turn on the live caption button located in the lower right corner. Today's session is being recorded. Recorded sessions are available in Teams on the Field Notes channel and the National Soil Survey Center's YouTube channel. Again, thank you for joining us. I hope you enjoy today's session. With that, I'd like to turn it over to Paul Reich to tell you a little more about today's webinar. Paul?
Hey, thank you, Casey. Today for our webinar number 13, I'm happy to introduce our two speakers. Our first presenter is Gary Harris. Gary is a soil scientist located in Robstown, Texas, and Gary will present his work on collecting data on urban soils to provide insight on how to improve soil health and water quality within the urban setting. Welcome, Gary.
Thank you. Just one moment while I get my screen shared. All right. My name is Gary Harris, and I'm the MLRA Soil Survey Leader in Robstown. And for the last couple of months, I've also been the acting MLRA Leader for the Rosenberg, Texas office. A couple of my colleagues, Travis up in Kerrville and John Wiedenfeld, before he retired from the Rosenberg office, helped me put together this presentation. And it's kind of a joint adventure, so to speak. All right, let's talk a little bit about urban areas within Texas. There's several urban areas within the state, as anybody might have guessed. There's several cities larger than 1 million people. The largest being Houston and the VFW, Dallas-Fort Worth Metroplex. San Antonio is a close third, and then we have the Rio Grande Valley that's over 1 million residents as well. There's also several other areas that are over 500,000 people. Austin, El Paso, Fort Worth, that resides up in the DFW Metroplex as well. And there's also several areas that are over 200,000 as well. So there's lots of urban areas within the state. So what has been done in the past as far as urban soil surveys? Well, not a whole lot. In the past, and at least in my MLRAs, the soils were mapped as urban land complexes or urban land undifferentiated groups, or even just urban land consociations. And the urban land component of that just had very minimal data, if any data, behind it. So there were very little information to gather in as far as urban areas were concerned. In my search for urban soil surveys of the past, I did come across this soil survey from San Antonio that was commissioned with the city and the uh, uh, Texas Experiment Station, AgriLife, uh, um, way back in 1964, if you can read the date there. But to my knowledge, this one is an anomaly, and I was unable to find any other um, urban soil surveys at all. So what are we doing now as far as urban soil survey and collecting data? Well, we have this little project that's occurring in San Antonio, ironically, and it's looking at the carbon content of the soil across the entire city. What's interesting about San Antonio is it covers 3 different MLRAs covered by 3 different offices. It's 83A, which is the Northern Rio Grande Plain, 86A, which is the Texas Blackland Prairies, and 81C, which is the Edwards Plateau. As you can see by the little map there, there's 59 sites that we've selected across the city to be sampled, and we're going to sample them a couple of different ways. We're going to collect samples and send up to Lincoln, as well as the Kerrville office has a MIR, mid-infrared spectrometer, to scan the samples as well, trying to help speed up and get a little bit more data quicker. We're also, as we sample, collecting dynamic soil properties, water infiltration, bulk density, those sort of things as well. So why is this really important? Well, the San Antonio Parks and Recreation Department are looking to ways to improve their soil carbon storage, increase the water holding capacity, and decrease runoff, mainly to help with flooding, and so that the city doesn't have to water as much for the ornamental plants around. And we're hoping that this will help other urban areas create a urban soil survey of the future. So you might ask, what else are we doing for urban soil survey? That project's fine, but we also have the coastal zone soil survey of West Galveston Bay. You might say, well, that's not very urban, but if you remember from the map, West Galveston Bay is just outside of Houston. They're the urban lands are encroaching and getting closer to the water every day. So what we're hoping is, well, we're in the final phases of this project. We're actually inputting all the data into NRCS right now, getting ready to wrap it all up. But we're mainly focusing on the subaqueous soils, but also updating the tidal flooding areas as well. This is a nice little picture of one of the past team members from the Rosenberg office collecting a sample off of a boat. Anyway, we have a couple of reasons why we're doing this project, and it's mainly to help for restoration projects of marine life. And as you can see, the buildings and houses there in the background, it's well within the realm of urban soil survey. All right, and these are the samples that have been collected across West Galveston Bay for the subaqueous soils and tidal zone mapping, and it's mainly to help control erosion and restore wildlife habitat. And again, this one hopefully will be an example that we can use further down the coast, because Texas has several other cities that are within the Texas coastline as well. So where do you think, where are we going with this? Yes. Well, with the concerns of urbanization and rapid growth in a lot of the cities in Texas, we're wanting to get the information that we can, and it pretty much as quickly as we can. There's already been some interest in a urban soils map in the lower Rio Grande Valley. It's one of the fastest growing urban areas in the nation right now. They produce irrigated and non-irrigated crops. The irrigated crops tend to be citrus, sugarcane, aloe. Oh, and the non-irrigated crops tend to be cotton and sorghum. But there's the concerns of increased water runoff and flooding, and really the loss of production of crops due to urban land conversion as well, for a lot of these areas. So with that, I know I went fairly quickly. Are there any questions?
Thanks, Gary. We'll give folks a few minutes to submit their questions in the Q&A panel. I actually have a question while we're waiting. In many cities, community gardens are becoming more widespread, and I was wondering, in San Antonio, are you finding that urban gardeners are interested in soils information, and do you have much interaction with the public in those kinds of situations?
Right now we're on the very front end of the San Antonio project, and there is some urban gardening happening in San Antonio, and there has been some interest from those groups, but right now we're focusing mainly on the parks, mainly because that's where we have good open access to collect the soils data at the moment. But yes, hopefully in the future we will be going to some of the private landowners in San Antonio to continue in the future.
Okay, I guess another question that comes to mind is, how much interaction do you have with the folks that are on the urban soil survey focus team? Do you, with Randy Riddle and on that group?
I do know that some of my colleagues have been in contact with them on a fairly regular basis, but I myself have not been in contact with them.
Okay, I'm not seeing any questions in the Q&A panel. But folks can definitely reach out to Gary. Gary has his email posted here, and again, really thank you, Gary, for this presentation, and hopefully in the future you and your colleagues can give us some follow-up information. Oh, I do see actually a question that just popped up. Let me go ahead and publish it. So the question is, can you talk a bit about the map unit compositions and soil series development?
Okay. For which project are we talking about? For the because with the San Antonio project, the project is in the very infancy, and we're not to the point of building new map units for that project yet. For the project in West Galveston Bay, we're creating an entire new legend for that survey area for itself, complete with subaqueous soils and tidal flat soils and the whole bit. As far as map unit composition of the past, a lot of these areas are 70–80% urban land soil and very little named soil.
And then I guess a related question is, what order surveys are these intended to be?
The original surveys in these areas were all Order 2. So these urban land soil surveys are going to be not quite in Order 1, but they're going to be a finer soils map than Order 2. So like a 1.5 is, I think, what we're shooting for as far as the San Antonio project. As far as the West Galveston Bay project, that one will be Order 2.
Okay. Another question that came up is, has there been any movement to prioritize known prime farmland in urban areas? These prime farmlands have been discounted and disregarded under the census definitions of lands. This is a national issue.
Not to my knowledge.
And also a follow-up to that, or what about soils of statewide importance?
To my knowledge, there hasn't been much done on that end of things.
Okay, I appreciate your answers, Gary. We've got some good questions. That's all I'm seeing for now. So I'm going to say thank you, and just let folks know they can reach out to you anytime to learn more about your projects. Appreciate it, Gary. Let me go ahead and share my agenda. There we go. Yeah, but, before I introduce our next speaker, I want to remind everyone that our next webinar, number 14, is planned for June 7, so keep a lookout for more details soon on that webinar. Okay, our final speaker today is Reuben Wilson from Raleigh, North Carolina. Reuben is a soil scientist, and Reuben's talk will be on using a soil survey approach to quantify dynamic changes in blue carbon. Welcome, Reuben.
Thank you, Paul. So yes, good afternoon and good morning from wherever you're at. My name's Reuben Wilson, and today I'll be talking about using a soil survey approach to quantify dynamic changes in blue carbon. This photo you see in the middle left right up here is similar to some of the environments we do work in. This is the Bird Island Tidal Complex in North Carolina, and just for scale, you can look down here and you can see our approximately 16-foot airboat versus this entire marsh. So just an example of some of the areas that we work in. All right. Taking its time, switching slides here. There we go. All right. So what I have in mind today is to give kind of a background information, understanding what blue carbon is, how we utilize the soil survey approach in it. And then we're going to do a short case study on what it looks like in the Albemarle-Pamlico estuary system and how it's been implemented. So first, our background information. So what is blue carbon? A lot of time when you start these conversations, people aren't exactly familiar with what you mean by blue carbon, but it's simply the carbon that's stored in these coastal and marine ecosystems. And it's both in the soil and in the living biomass. And the coastal and marine ecosystems that we focus on in particular are those with seagrasses, salt marsh, estuarine mangroves, oceanic mangroves, as well as freshwater tidal forests. And based on this graph, you can see here from Murray, BC et al., you can see that these blue carbon environments, estuarine and marine, contain significantly more carbon than your average tropical rainforest. So they're these really carbon-rich ecosystems considered this blue carbon that we're trying to understand better. So why map it? Why does blue carbon matter? And why should we use a soil survey approach? So just like any other soil property, it's an inventory. We're trying to gather a better understanding of it to help us inform our management decisions and better understand the environment and its functionality. So we want to understand where this blue carbon is highest concentrated, how it's affecting its environment, how is it changing with climate change. There's a lot of demand for blue carbon information pertaining to climate change and informing those decisions as well with climate banks. And you know, it reminds me of something, the original soil survey. You know, we're just going back and going to these areas that were initially glossed over, weren't mapped particularly well for any number of reasons. And we're doing our due diligence this time, and one of the things we're understanding better is blue carbon in these ecosystems. So why is the soil survey approach best? So as we understand at a base level, it's the ability to inventory a large area with appreciable accuracy. So you're going into these landscapes, you're identifying your landforms, you're correlating soils, you're seeing which soils occur where, and we're using that information to help us map carbon. So you're creating a carbon map based on the soil map like you would with any other kind of characteristics. And the soil survey approach is also best because it goes to 200 centimeters. So in the world of soil carbon mapping, there's a lot of disagreement or discontinuity with people understanding and how they want to sample for carbon, because certain individuals will just sample the top 50 centimeters, they'll sample every 2 centimeters, they'll sample every 15 centimeters. People really don't have a good concrete way to like scale up carbon data across a landscape. They can only scale it at that one place where they sampled at. But when you use this soil survey approach and you sample on a horizon and you sample to 200 centimeters, and you can tie this information to these landscapes and landforms, and you can tie them to these soil units, you can really generate a blue carbon map going across an entire area. And so it's also important to go 200 centimeters. If you look at the two soil profiles on the right of the slide, Appoquinimink and Transquaking, you see why it would be important to go to 200 centimeters for this blue carbon, because the first 76 centimeters of Appoquinimink is mineral material, but then you have 100 centimeters of that organic material, that really rich blue carbon that we're trying to capture in these soil maps that would be not accounted for whatsoever if you only went 50 centimeters. Likewise, with the other profile on the right, if you only went 50 centimeters, you would only catch a third of the carbon or the organic matter in this horizon. So we want to go that full 200 centimeters and capture all of this blue carbon information. And beyond that, it's an accepted national standard. The soil survey approach is what we've done for over 100 years. It captures the soil profile in perpetuity and the properties that we're interested in. So it's really just the best way to go about capturing these properties on the landscape. So now that we've covered the background of what blue carbon is and why we use the soil survey approach, to understand it, I'm going into a case study of where we've done this in the Albemarle-Pamlico estuary system. So if you're not familiar with the area, the Albemarle-Pamlico estuary is in North Carolina and Virginia. It's actually the second largest estuary on the eastern seaboard behind the Chesapeake, and it drains a large watershed, very mixed use, urban, whole lot of agricultural land, as well as forested land. And so what the research design or the mapping design to look like was this. So if you look at the left side of the slide at this map, you'll see green dots, yellow dots, or yellow triangles, and red squares. And so what these represents are different ecosystems along a salinity gradient in this ecosystem. So we sampled 12 sites across a salinity gradient on the Albemarle-Pamlico estuary. And so the green dots up here are your tidal fresh, and this is what they look like. These are your traditional hardwood, upland, swamp, forested areas, bald cypresses and water tupelos. And then as you move down in the sound, you get to these yellow triangles, which is our ghost forests, which were historically fresh, but have since received flushes of saline water from either sea level rise, wind-driven tides, hurricanes, anything that's gave it a flush of salt water that stressed this freshwater vegetation to the point to where it can no longer survive and it starts to die. So this is a good example of it here. This site is right at this yellow triangle. It's Alligator River. And then finally out here, the traditional salt marsh. So these are mesohaline, high salt content. You have your salt tolerant species out here. So this picture in particular is down here at Swan Quarter National Wildlife Refuge on the estuary. And as you can see here, it's just several miles extensively of marsh material. Black needle rush is dominant, a halophytic vegetation right there. And we cover this entire scale looking through this peninsula doing our blue carbon inventory. So what we did at each 12 of these sites was we developed a mapping timeline. And so what I mean by a mapping timeline is we simply performed a transect with 3 sets of cores. We did a subaerial core, which that would have been approximately 25 feet from the shoreline. Then we did our nearshore core, so that would have been a subaqueous core taken to the utmost closest edge that we could get the coring vessel to the edge of the shore. And then we did our offshore core, which was far enough offshore that we believed that we were on a different map unit or beyond the historic soil mapping polygons of the 1980s, specifically for this area. So if you look at this progression of satellite photos up here, 2017 in the bottom right's probably the easiest to explain it because you can see here your subaerial is this red. So in 2021 when these cores would have been taken, this was actually underwater. And then this green out here, our offshore, is what we believe to be beyond the traditional polygon lines. But it actually turned out that this site was rapidly eroding so fast that it was still within the polygon. The actual polygon from 1987 was right out here. So that just shows how rapidly some of these areas along the coast of North Carolina and the Albemarle-Pamlico are eroding. And this site in particular, one of the reasons it was eroding so fast is it's about 18 kilometers west of the Oregon Inlet from open tidal influx. So mapping at depth, I kind of touched on this earlier of why it's important to go to 200 centimeters with a soil survey approach. This graph from Gorchinski 2022 really puts it into a scientific perspective. Here on the left side, on the y-axis, you have your soil organic carbon pool, so your amount of carbon that's in that soil, and then on your y-axis you have that, and on your x-axis you have your average water salinity. And so as you go from left to right, your salinity is increasing. And so graph A is only to 100 centimeters, while graph B is only to 200 centimeters. And really the take-home story with this graph is simply that if you only go to 100 centimeters, you underestimate the amount of carbon that's in marshes and the ecosystems as a whole. But it also gives us some hope, because if you look at this line here, kind of bending back up towards the end, it shows that as we go from this transformation from fresh forested to ghost forested to marsh, we might actually be accumulating carbon over time as these ecosystems recover. So that's just an interesting conclusion that could be drawn when you go 200 centimeters, but if you only went 100 centimeters, you wouldn't see. Another reason to go to 200 centimeters, these are some of our hidden gems. So over here on the left, this was in a tidal freshwater site. So one of the green circles, the upper 50 centimeters of this soil was just alluvial silts from the mouth of a river. So if somebody came in here and only sampled 50 centimeters, they would not have captured any real appreciable carbon from this profile. But if you go to 100 to 150 centimeters and on down to 200, it's actually old sapric organic material, very decomposed, carbon rich, that wouldn't have been accounted for. Likewise, this is from a offshore core in the same transect. And if you only went to 60 centimeters, it's just coarse sands and there's no real carbon to speak of. But once you go beyond, try to go to 200 centimeters, you start capturing this old red organic material from historic bald cypress swamps in the area. So it's really important to go to 200 centimeters for people trying to understand blue carbon. Another thing that to understand with these transects as we build them is the historic rate of land loss in the Albemarle and Pamlico estuary. So what you're looking at here on the left is your rate of land loss, which is meters per year. And down here on the x-axis is a rolling 10-year average. And really the take home to see here is that originally ghost forests were eroding upwards of 2 meters a year, so 6 feet. That original site I showed you, where we thought we were entirely off the polygon unit in 1987, has actually eroded approximately 300 meters over 35 years. And it's really staggering to look at how much land is being lost in these really carbon-rich ecosystems. But also here we have some hope, because we see that our marshes in the past 10 years-ish have been able to keep up with sea level rise and erosion. So that is promising to see. So when you look at these large landscapes, it can be very difficult to visualize the erosion and understand the land that you're losing, the carbon you're losing, the soil you're losing to sea level rise and other erosional factors. So this is a freshwater tidal forest. This is at Roanoke River. This is one of the green circles. And what the historic shoreline would have looked like is where you see these lingering bald cypresses out far in the water. Oh, pardon me. And so traditionally that shoreline would have been out here, but over the past 50, 60, 70 years, the water has gradually moved in. Trees have been thrown out of their shallow rooting depths, buried, carried off, and it's slowly moving the shoreline up. So this is what erosion looks like in this tidal forested area. Likewise, this is a ghost forest. So these are these areas that receive that saline flush of water that's enough to stress the freshwater vegetation and kill it. If you look down here in the water, you can kind of see these shadows. These are old stumps from bald cypress trees that we saw on the last slide. And you can see you have these dead stands of pines or cypress or tupelo trees that have tried to survive but are unable to due to the high flush of saline water. And so this is just slowly eroding back and killing this vegetation as you see here. And that would be, this is what the erosion looks like in a ghost forest. And so this is visualizing the erosion in the marsh. So what I'm standing on here is actually the mineral platform of a marsh. So sometimes marshes form with, say, if it was a 200 centimeter profile, the bottom 100 centimeters may be mineral and the top 100 could be organic. An example, that's what we see here. And the wave action, hurricane, storm surge is gradually eroding all the organic material off the top of the mineral platform in this environment. So I'm standing in about 2 feet of water right here, and pretty much to the edge of this image here is where organic material has been eroding over time in marshes. And so to help us better understand how much blue carbon, how much erosion is occurring on the Albemarle Pamlico estuary, I did a NDVI, which is just a way of separating land and water using ArcGIS and rasters. And what I did was I separated the land from the water, and over a period of years, I calculated how much less land in pixels there were over time. And really what the purpose of these next few slides is to show you the certain ecosystems we talked about. So the ghost forest, the tidal freshwater, and the marsh. So right here, this is the area we're going to look at. And what you're seeing here, this purple, red, yellow kind of tinge of color is land that is now water that was occupied in 1984, I believe, by surface. It was surface, it was swamp, but now it's a ghost forest that's casually been eroded in. And so what does that look like on the ground? What does that look like in our transects when we create our mapping timeline? And this is the interesting part. So you had this ghost forest soil. This is your subaerial core A. So over time, as that organic material is eroded, as the sea levels rose, as it's got these flushes of salt water, that organic material is eroded and submerged that soil. And so it looks like this. You can kind of see the similarities here. We have some difference in colors due to the accumulation of sulfides. And then over time, on a longer period with more erosion and longer exposure underwater, you have the rest of this organic material on top erodes into these old fluviomarine and marine sands. And so the statistic we're looking at over here on the right, the peat loss upon submergence, is the amount of peat or organic material that's lost when these subaerial pedons become submerged. And you can see it's virtually all of it in ghost forests and marshes. So likewise, we're going to look at marshes. So this area down here near Swan Quarter, this green, once again, these were areas that were historically land back in the 1980s, but due to factors like tidal incising, rack lines collecting and killing vegetation, they're now considered water, open water habitat and spaces in 2021. Just as well, this area over here is the one we showed earlier, Point Peter. You see this blue, and I mean, it's very pronounced how much land has been lost on the east side of this peninsula since the early 80s right here. We're talking 300 to 350 meters. And these are what those soils look like in those marsh. So you kind of have this difference of soils here on the subaerial, this estuarine environment that has these depositional patterns. So you get a lot of like kaleidoscope soil colors of estuarine environment. But as soon as it becomes submerged by erosion, storm surge, this organic material gets washed away and it's eroded. And then over time, further out, you completely lose all of that organic rich material on the top. And finally, the tidal forested area, right up here at the top, this is the mouth of the Roanoke River. This is going to be from that same photo I showed you earlier of all the bald cypress trees. And you can see really you're just seeing some erosion right here on the edges, primarily from sea level rise. And that's the factor we're seeing in tidal forested. And this is also an interesting mapping transect here between your subaerial, your nearshore, and your offshore. So this is actually at the mouth of a river. It's almost like a subaqueous delta. So as you move forward out into the river, you actually have a decrease in the water level. So it goes from 188 centimeters of water to 134, and you have this mineral soil out there. But the big thing to keep in mind here is you have this erosion from the subaerial to the near shore. And if you were to stack these two ped-ons side by side, they really don't look that different. You have a thin mineral cap, and then you have buried organics. But actually, you've almost lost 188 centimeters, nearly an entire ped-on's worth of carbon. And that just speaks to how much carbon's actually in these ecosystems, the tidal forested ones, to extreme depths. So it's really important to make sure you get your elevation corrections, but also you're going your 200 centimeters to understand how much organic material is really here and how much we're losing. So this is just kind of sum up here how much carbon is being lost in these ecosystems. So we visualized our erosion and our submergence by building our timeline with our subaerial, offshore, and nearshore core. And we can understand that the average in the environments, the 12 environments we saw across the entire salinity gradient in the Albemarle Pamlico, at the least in marshes, we're losing 471 megagrams of carbon per hectare. A megagram is approximately 1 metric ton. And it only goes up from there, with freshwater forests losing upwards of 730 megagrams per hectare. And so the data presented in this paper was pulled from, or this PowerPoint was pulled from Gorczynski and Ricker in 2022, as well as some of my own work with Dr. Ricker at NC State. So thank you for taking the time to listen to my presentation, and I'll be happy to answer any questions.
Hey, thanks, Reuben. Appreciate that. Yeah, we'll give folks some time to type in their questions in the Q&A panel. Yeah, I'll just mention I didn't realize how much carbon is stored in these deeper soil horizons in the coastal zone. That was definitely an eye opener. You know, I've done a lot of past work with global estimates of carbon stocks, and I know that often those stocks are reported to 100 centimeters, and I know like the FAO, they have a pretty good global soil organic carbon map, and they report the stocks only to 0 to 30 centimeters. And I guess that's okay for terrestrial areas, but certainly your presentation shows that that's definitely not adequate for the coastal zone areas, where you definitely need to be collecting and reporting to that 200 centimeter depth. So that's definitely, your presentation is really eye opener for me. I'm sure it is for a number of other folks.
Right. Yeah, absolutely. And there's some of these places where these are very stable environments these tidal freshwater swamps have been, and the carbon goes several meters, or the organic material goes several meters deep. So it's pretty impressive for sure.
And then also, certainly another thing that really comes to mind about your presentation is just really truly how dynamic these areas are, being especially subjected to hurricanes that come through, and all the changes and with global change. And so that's a whole other area that's pretty important for us to be working on. So I really appreciate your work on that. I do see one question that's come through. Says, I recall from your last presentation that there was no definitive answer to show where the carbon is lost to. Why is it considered lost rather than deposited elsewhere?
Right. Yeah, and that's a great question. And I always tell people that's somewhere where there definitely needs to be more work in this research, because this was kind of scratching the surface of understanding the dynamism of these systems and what's happening as they submerge. I think for the purpose of this work, we considered it lost because it was no longer present on the transect in any of the environments we worked on. So if you'll recall, in all 12 sites across the entire salinity gradient, we didn't move further in and see huge levees of organic material that had been deposited on the surface from wave action, and you move further out and you don't see any organic material at all. So based on the parameters of the project, it's the safest and most logical thing to assume that it's just not accounted in the system for anymore. But that's not to say that it isn't, but I think scientifically you soundly can assume.
We'll wait a few more moments to see if any other folks have any other questions. I don't see anything else in the Q&A panel. I couldn't remember, Reuben, if you mentioned the actual status of the soil survey in this area. So is it in the middle of the project, or is it getting near completion? What's the status of the survey for this region?
Right. That's a great question. So this initial project that I worked on, and also Lori Gorzinski, who's just recently came on with the agency in Richmond, was more of an inventory to understand what soils actually looked like out there. Probably in the next year or so, we're going to start initiating a soil survey or a subaqueous soil survey in Currituck Sound, which is part of the Albemarle Pamlico estuary, but it's just up the way. So we're probably looking at starting to map these areas and develop these carbon maps that we've prospectively talked about in the next probably 3 years, I would imagine.
Okay. I do see another question that just came through. If going to 200 centimeters is important, why not 300 centimeters? How can we make a good call on what depth to stop at with such deep, organic, rich soils like these?
Right. Yeah. I mean, that's another great question. Like I said, these systems can go several meters down, but you usually don't see that extreme depth of organic material except for in these tidal forested systems that I understand. Now, if someone was to come to me and say they wanted to take a soil survey approach to run a 400 centimeter transect to understand and capture all these organic materials, I would say, great, I would love to, but the amount of hours, equipment that it takes to conduct a survey to that level, that's really a lot of the pushback that we get for going 200 centimeters is the amount of work it takes to get 200 centimeters. So in theory, I would love to map it till I hit the bottom. But it really becomes an issue of equipment, hours, and the ability to do the work on a realistic time scale that helps us choose the 200 centimeters is one of the best, as well as it being with the traditional soil survey approach of being able to encapsulate those properties.
All right. Not seeing any additional questions. Well, I would really like to thank both of our presenters today. Thank you, Gary. Thank you, Reuben. And also thanks to all the attendees watching today. Everyone have a great day, and we'll say goodbye for now. Thank you.